Welcome back. SignPost has developed an accurate breast cancer screen which does not rely on an image reader. Now, to find out more about this innovative technology, we've got the CEO and founder of SignPost, Peter BLANEY, with us, and also Family Office Insight CEO Arthur BAVELAS. Great to have you both.
Thank you, Jane.
So, Peter, how does BreastDefense is the name of the technology that your company SignPost has developed, right?
Right.
How does that work?
It's a molecular diagnostic. It relies on specific addresses being activated on the epigenome, which is the dust that settles on your genes. And so it turns genes on and off. And we found a small set of addresses that are virtually, well, it's 99.9%. If you have these addresses activated, you have invasive breast cancer, regardless of the subtype, regardless of the stage.
So, don't need a mammogram. Well, how does the test go? Is it a blood test?
It's a blood test.
Okay.
But yes, you
still will need a mammogram.
well, there's
okay.
going to be, we're not trying to displace mammograms. We're trying to assist.
Okay.
So if you've got a mammogram and there's a suspicious lump,
I see.
there's still a hard time determining whether or not that lump is invasive breast cancer or benign and can be left alone.
So another diagnostic tool to help find breast cancer.
To give you certainty.
Okay. Okay.
We answer a really simple question.
Yes.
Is this invasive breast cancer or is it benign? And that's the question that the pathologists and radiologists and oncologists and surgeons have told us over and over again that they have the most difficulty determining in many cases.
Okay. And you also have been a winner with Arthur's Family Office Insights group for your business. So, Arthur, what was it about SignPost and their new technology? I mean, this seems like revolutionary stuff.
So the Family Office Funding Challenge, which is the component of Family Office Insights where entrepreneurs compete, I will admit that Peter and his group came to the top with a big margin away from everybody else. And there's lots of metrics that we use, and we don't have time to go into now how that happened. But clearly, if you could, and one of the things that I was interested in asking you is that wouldn't it make sense to do it the other way around once this is perfected, to do your test first and then do the mammogram second?
Well, I think that is, in fact, what will often happen. The mammographers, if they get the results from our test, they will find where the cancer is. It will be much easier for them. So I think they're quite complementary.
Does it detect all kinds of tumors, or?
It's, at this stage, we're focused on breast cancer,
Okay.
but it will detect any subtype at any stage from 1 to 4.
Okay. And so then my question is, why isn't everybody using this yet?
Because it hasn't been completed. We're
Okay.
raising the money to validate the
I
test
see.
now.
Okay. So you need to scale and.
Yes. So the normal procedures that you have to go through to validate a test, we're completing.
What will that take? Is that an FDA approved thing, or?
No, this is a CLIA test, so it's under different legislation, so it's not under the FDA, although the FDA does watch what happens with CLIA tests. But almost all of the new molecular biology tests that come out are LDTs, they call them lab developed tests, and they work under CLIA legislation. So it can get to the market much faster and faces less regulation than the FDA.
Interesting.
So what's much faster mean, for example?
Well, it means, half a year instead of 3 or 4 years.
That's much, much faster.
Oh, yeah. And you're also using big data to help with diagnosis. How does that fit?
Well,
into
that's
this?
how we got the addresses that we need. There are 6 billion nucleotides in human DNA, and how do you find the right addresses in 6 billion? It takes big math, and fortunately there are huge databases out there now, like the Genetic Data Commons and so on, and the Human Cancer Genome Atlas, where thousands and thousands and thousands of people have had their genomes fully sequenced, and these are publicly available databases that any researcher can make use of.
Should we be optimistic about the application of this type of technology that you discovered and the ability to cull through these databases for other, what do you call them? The nucleotides for other problems?
Absolutely. What's really come about over the last few years is that what they call next-gen sequencers. It used to take a decade to sequence a human genome and cost billions. Now you can do it for under $1,000 and you can do it under an hour. And so this stuff is really, it blows Moore's law out the window. The advances that have been made in the human genome are unprecedented in the history of science.
So walk me through the process. Let's, like, say in 10 years, and I go to see my doctor for an annual exam. How do you envision this working, and how is it being implemented into offices? Wow,
when you say 10 years, that is so far out.
Okay, 5. 3.
Okay, 3 years. 3 years I can handle. 10 years, it's too far. All the platforms.
There'll be so many more developments in 10 years.
There's
amazing.
Absolutely.
Okay, 3 years.
So in 3 years, I think what will happen is there will be blood tests available, blood draws that people will take, and you need a fair amount of blood, maybe, the standard vial. You need a full vial, and you'll exhaust that vial on probably one test. But there will be multiple tests out there for all kinds of diseases.
I have a daughter, so it's wonderful to see what kind of world she's going into. So thank you so much,
Yeah,
Arthur.
we're super excited.
Yes.
Thank you.
And thank you as well for joining us. We'll be right back.